So good morning, everybody. Thanks for coming. Chapin is a postdoc in my lab at York University, and he'll be speaking to us about some projects that he's been working on recently, specifically to do with in-host modeling.
Thank you, Jane, and thank you very much for the invitation. Uh, oh,
This is, I think we have to start first with the land upliftment and then we move on to the speaker.
okay. Sorry about that.
Please, thank you. We acknowledge the land OMNI-REUNIS network's head office is located on York University, recognizes that many indigenous nations have long-standing relationship with territories upon which York University campuses are located. That precede the establishment of York University acknowledged its presence on the traditional territory of many indigenous nations. The area known as the Toronto has been caretaken by Anishinaabeg Nation, the Budaksi Competency, and the Flora Weta. It's now home to many First Nations, Inuit, and Metis communities. We acknowledge the current territory holders, the Mississaugas of Great First Nation. This territory is subject of the District 1 Bill Congress, an agreement to peacefully share and care for the Great Lake regions. And
So thank you everyone for joining us today for the OMNI Super Spreader Seminar Series, which is happening every month. And we have so many talented speakers throughout the year, and we will continue with this seminar series every month. And you are always welcome to register and attend. And today we will have a talk by Dr. Chapin Korosec. And Dr. Chapin Korosec is a postdoctoral researcher at the Modeling and Immunity Lab here at York University, who is supervised by Professor Jen Hassanam. So I'll ask Professor Jen Hassanam to introduce the speaker, and then we can continue.
All right, thank you. I thought I already did introduce the speaker. So welcome everybody this morning. Happy to introduce Chapin, Dr. Chapin Korosec, who is in my lab, and he'll be speaking on in-host modeling.
Thank you, Jane, and thank you very much to the presenters for the invitation. Excited to be here virtually. So today, I'm going to talk about the research that we've been doing in the group, in the Heffernan group, particularly around modeling the immune system and specifically responses to vaccination. And so I'm going to talk about a lot of different stories and papers, but there's all going to be a common theme of dealing with basically the lymph system and how it responds to perturbations. Because I often run quick on time or late on time, today I won't, I promise you. I will start with my acknowledgments. So of course, all the work that I'm talking about today, Jane Heffernan at York is the PI, and we've also worked closely and collaborated with a lot of different researchers from around Canada and the States. So Morgan Craig, Ian Moyles, Lindy Wall, Jessica Conway, James Waltmau, and Mario Ostrowski, and a few postdocs in the lab were involved in previous projects. That's David Dick and Susan Farhang-Sardrudi, who have moved on from the lab to do other great things. And we have a close collaboration with the NRC. Specifically, our first few papers were with James Oy and Sajad, and now we're working closely with Sajad on some of the machine learning stuff. So every time you see machine learning, just know that there's a lot of work and supervision that Sajad has also been doing behind the scenes. So some quick facts about the lymphatic system. These are just some of the things that I have gotten excited about learning, not necessarily what we've used in our models, but it's just such a fascinating system. So our lymph system runs in parallel to our circulatory system. And obviously, we are aware of our hearts beating and pumping blood throughout our bodies, and that blood is distributing all sorts of resources and oxygen to all of our organs. But our lymph system, which runs in parallel, does not have a heart to push all of the lymph fluid around. It relies on, has a loose coupling to a heartbeat, but it really relies on our movement and coupling to our kinetic motion to push that lymph around. And so each of us have approximately 600 or 550 lymph nodes, but there's a distribution across a population of people, about plus or minus 200 lymph nodes. I'm not sure why. I'm interested in learning why, but there's a huge distribution of lymph nodes in all of us. despite them being a really important quantity, a really important organelle, they're not conserved in their absolute number. They're coupled via a network of lymph, as I had mentioned. Lymph flows at about 1 to 10 millimeters per second, depending on if you're moving around a lot or relatively stationary. The immune system as a whole is comprised of approximately 1.8 times 10 to the 12 immune cells. And all of these cells form our complex immune response. That's all these complex T cells and B cells and NK killer cells that are comprised of our immune system. And together, the system is capable of memory. So when we get sick, we often remember that antigenic response. It's capable of non-self identification, which might be the most fascinating aspect of our immune system. The fact that it doesn't consume itself and it can respond to external invasive pathogens, and there are diseases involved with the non-self process going awry. And I'll talk about a few of those in the context of vaccination. And what's really interesting, and a lot of work done in the early 1980s by the group or by Alan Perelson, is that it's been shown to consist of a complete repertoire. So a genetically, we'll say, healthy individual has a complete repertoire, which means that any form of antigenic response, any peptide form, our immune system has an ability to match that form with an antibody response. So it's mathematically been shown to be a complete repertoire or a complete system for a genetically healthy individual. There are genetic diseases involved in incomplete repertoires as well. Very complex, highly heterogeneous. We'll talk a lot about that. And many aspects of the lymphatic system, irrelevant of the perturbation that's being received. So the holistic state of our physiological function, a lot of those facts remain unknown. A really cool paper that came out a couple of years ago now is about where do even plasma B cells perform their function. It's not in the blood. Is it in the lymph? Is it in the bone marrow? And if so, where in the bone marrow? And so we're still learning about fundamental foundational facts of our immune system. And so it's a really cool area to be doing research. We're going to be talking about our projects that have to do with what happens when we receive a vaccination, what happens through time, and can we learn about the intricate components and interdependencies that form through time and decay with time in our immune system. And that's the main function. And so basically, we receive a vaccine. If it's in our deltoid, it's in our muscle, and it's in our muscle because there's a lot of blood flow. And so all that antigen is picked up and distributed likely to the nearest topologically associated or lymph system. And so likely these are the lymph nodes that we're really going to be focusing on. Although in our modeling, we model them with a sort of a net response. I'm not going to do a talk about any network modeling today. And this is just to set the stage for what we're going to get into. Theoretical immunology, so how I view myself. Basically, we aim to develop predictive, statistical, and computational models to gain deeper insight into the principal components that govern immune system function. So we work very closely with clinical immunologists that are coming up with studies to unravel mysteries of the immune system. And from a theoretical point of view, we want to do models and things to gain deeper insight and understanding of data sets and immune system function. And so to get a little bit deeper, to set the stage for our first model, set the stage for our first model, we receive a vaccine and we want to know what's going on in our lymph node. Our lymph nodes have an organizational structure. They have the yellow cells, yellow and white cells or white blood cells, and the red sort of webby cells are a different kind of white blood cell. And they form these domains and they surround these subcompartments of naive plasma B cells. And if you recall, it's plasma B cells that eventually get trained to produce antibodies. And so with this structure in mind, we want to create our first goal was to create compartmental modeling to understand net responses. And so, for example, this is not my figure. This is a nice figure that I'm taking from a different paper. A way to think about this is although we have, say, 600 lymph nodes or 550 lymph nodes, and there's lots of different B cell compartments and germinal centers within all those lymph nodes, you can think about modeling the net B cell response, the net ability of B cells to be trained against a certain pathogen and produce antibodies. And you can think of coupling terms between B cells that are required physiologically. A B cell needs to physically touch a certain subtype of white blood cell to get that information and use it in a useful way. And you can think about coupling to other compartments and modeling those compartments if you have the data to support an informative model. And this is how we're thinking when we receive a data set, or we're talking about clinicians that have data. We're thinking about building these compartments to learn something useful that you cannot learn from statistical modeling, for example. In general, we're interested in immunological trends as a function of various different stratifications. We'll recall, so my research started during the COVID pandemic, where the hot topics were like, what is Moderna a lot better than Pfizer? Is AstraZeneca not a good vaccine? And all these sorts of things. We're really motivated to pick out differences in magnitude of response and differences in time scale of response between different types of vaccines. We might be interested in sex, depending on the context of the study. Is there a meaningful difference between male and female responses or sex at birth responses? Age, immunosenescence. As we age, our immune system has dysregulations that just emerge through time that make it typically are correlated with less antibody responses. Does that matter? Is something that we're interested in. Various different comorbidities. I'll focus on HIV and a little bit on asthma today. Comorbidities can manifest as various different types of dysregulations in our immune system. And of course, something that we're all been made very aware of in the COVID pandemic, dose-dependent dynamics. Do you need a certain number of doses to bring your immunity to an optimal level, and what is the dosing interval perhaps to maintain that immunity and maintain protection? These are all different things that we consider in our papers, and I will hopefully have pulled from the most interesting of these things to tell a cohesive and not overwhelming story. So let's begin with how we think about developing, thought about developing our first model, which is a vaccination model. And so, for example, this is a model to understand mRNA therapeutics, specifically for the COVID vaccine. So these are the Moderna and Pfizer vaccines. And so we have some sort of a variable to guide our initial conditions. So a certain amount of vaccine that is delivered to the body that initiates an entire cascading process of immune function. That vaccine has a loss term. There's a certain amount of vaccine that is picked up by the blood and it's drained and processed by our liver, and it does not elicit an immune response. And there's a certain amount of vaccine that just does not cause an antigenic response. It's not picked up by our professional antigen cells, and we model that with a gamma L term. But there is a coupling term of the vaccine that causes a vaccinated cell response, and these vaccinated cells, if you're familiar with the physiology, are like professional antigen presenting cells. They're dendritic cells. Subset of them are macrophages, and their goal is to take this information to our lymph nodes where all this complex magic happens. And we model that compartment just as a vaccinated cell compartment. And we're going to benchmark vaccinated cell dynamics with other mammalian studies on mice where we're allowed to do luciferase assay studies. When we get to that, it's an identifiability problem. And then there's just a half-life associated with vaccinated cells in the body before they lose that information. And that's gamma V. And so basically what we want is some sort of initial compartmental understanding that leads to a perturbation to a lymph node that is initially naive to that antigenic response. And the outcome is a humoral response, humoral meaning just the suite of antibodies that are now going to be in circulation in that person. And there's also the other component of the immunity, which is the cellular immunity. And our data, the easier data to collect in humans through time that we work with, that the clinicians have carried out in these studies, are collecting that humoral data and collecting that cellular data. So those are the data types that we're going to use to benchmark or fit our study. And then, of course, if you're familiar with fitting studies, then a lot of work is gone into how does that information flow through the model, and how well can you practically confine these compartments that you're really interested in learning about. And that's where a lot of the time comes into, and that's where a lot of my talk will now diverge to. And so the full model, if we consider, oh, sorry, there's a video. I just need to exit this for 1 second and close my video because I need to see these compartments. The full model looks like this. So we have a lipid nanoparticle leading to vaccinated cells. Vaccinated cells then enter into the lymph nodes and prime two different types of T cells, CD8 cells, and CD4 cells. CD4 cells are a cell type that passes the information on to a bulk plasma B cell response. So this is net across all lymph nodes, and then they produce antibodies. And then the antibodies are what we want to fit to. All of these cell types within our lymph node are constrained and controlled by what's called cytokines. And so these are cells, and cytokines are molecules or small proteins. And cytokines are involved in a number of different feedback loops to turn up or turn down responses within our lymph nodes. And so we model cytokine. We have cytokine data to use in our fitting algorithm, and we're using those cytokine data sets to try to understand the influence of cytokines within lymph nodes on these cell types, CD4 cells and bulk B cell responses. And cytokines have their own associated clearance term or degradation term, or loss term, I guess, whatever. They're removed from the system at some rate. So this was our first model that we published in scientific reports. And oh yeah, I wanted to mention, I highlighted at the very top there gamma V. And so early on when we were looking at the structure of this model, gamma V turned out to be a highly non-identifiable parameter. It's very far away from the data that we're fitting. So it's intuitively non-identifiable, I guess you could say. But it is an extremely important parameter to get right, or to get close to a physiological correct estimate, because it guides the entire time scale that cascades into all of these interesting dynamics that we want to benchmark for people. And so we had to make a decision early on, and how we're going to fix this. And luckily, there are a lot of researchers who have been interested in absorption rates of vaccinated cells and the time at which in the intramuscular tissue from the mRNA vaccine that's translating mRNA into proteins. How long does that occur at the site of vaccination? And there's a lot of mouse studies on this. There's a particularly fantastic one that actually has timescales associated with it. And you can see in here, for example, this same mouse on the left is the mouse on the right. And they are looking at heat maps of activity of translation. And we use that information to put a half-life on at least mRNA vaccine translation half-life in mice, which are mammals. I know mice, a lot of people say mice are not people, which is true, but we're using this data from mice as a way to fix this parameter, which matters a lot in this model. Okay, so our first set of results, or I'm going to go through a few highlight results from this paper. We published it in Scientific Reports in 2022. We were one of the first people on the archive, actually, to put a number and a confidence interval on a decay rate. However, that was in 2021 of October. We had an antibody decay rate following 2 doses of Moderna and Pfizer, but then we were stuck in review limbo for a year. And so it came out, as like paper 12, I think, that had some sort of antibody. But I like to say that we were pretty close to the beginning, and we were really close to what is accepted values, which is really cool to see. And so basically, we're interested in age, and we found a statistically defendable result that younger individuals age 18 to 55, that's just how the data was categorized, produce a slower decay rate. And then we were able to show that there is a faster decay rate associated with increasing age It's very shallow here on this plot. And so that was one of our initial results. So that was nice. And to be more precise, we wanted to look at the benchmarks of their percent categories of loss. So we don't just want to put like a half-life on decay loss as a function of multiple doses. We wanted to benchmark that loss as a function of percent. Because at this time, a lot of the literature was around what's the correlative protection. We don't know, but we can correlate percent loss with people that are now getting reinfected and ending up in the hospital. And that was the way we took this story. And so this plot is predicted immunity remaining. So that's percent of peak. So we take the peak antibody response. We turn that's now our max. And then we renormalize the plot as a function of percent of the antibody in circulatory system that's been modeled as a function of time since dose 1. And when we were doing this, this was because the initial CDC recommendation for dose 3 and it was 270, ended up being 270 days post dose 1. And we were like, whoa, 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 we don't know what the correlative immunity is, but surely with 99.7% loss of peak immunity, you're now not going to be able to fend off a new infection. And that turned out to be correct. And the CDC, not because of us, but because people were rediscovering this result at the same time, revised their plan to have it 6 months post dose 2, which lands you in an area of about 70% loss or 80% loss post peak. And so there is something to do with correlative protection there, although we weren't able to benchmark the exact or benchmark an actual value for that. And so that was one of the main sort of hot topic things that we were working on at this time of the pandemic, and this is how we told that story. And then this is the Moderna plot. The Pfizer plot looks really similar to this. And then, yeah, this is old news now, but we found like the antibody peak post-dose 2 occurred 61 days post-dose 1. So basically, we received dose 2. We were told to get the second dose 28 days post-dose 1, at least in the United States of America and Canada. There's a distribution of that. But everyone was told to get the second dose 28 days post-dose 1. But the peak in antibodies doesn't occur until 61 days post-dose 1. And so there's a lot of interesting dynamics going on under the hood that are driving antibody profiles long after, 30 days after dose 2. And this was also found to be true by a number of different studies after we had published it. And one study, when we published it, that we cited, which says 57 days, and we found 61. So it is always nice as a modeler to be to have results confirmed by others, because obviously there's always this feeling of hopefully we did things right, and I think we did. And so another highlight is we're interested in the cytokine feedback loop. It's hard to understand that clinically, and basically there's this paper I alluded to that found long-lived plasma cells are accumulating in bone marrow. But what's really cool about this paper is they also benchmark plasma B cell timelines. And so, for example, in mice, this is a mouse paper that we're going to refer to. But in mouse, they found a bulk plasma B cell cessation half-life of 23 days in mice that are inoculated with a model antigen, meaning that it's a vaccine, but it's a vaccine where it's designed so that they can easily pull out time-dependent information. They're not inoculating with like influenza or a type of actual virus or it's a model antigen. And so in our study, we found with the error on our B cell compartment that in people, the half-life ranged from 7 to 20 days. So we can't tell. It's an equivalent metric from 7 to 20, but it was really nice to see that our best fit results were within I would say a ballpark range of what's found in mice. And again, there's no reason to think that a mouse with a far smaller and less complex and I think 3 orders of magnitude less total cells would have the same time scale, but it's nice to see that we were close. And so I thought that was really exciting, and we dedicated a couple paragraphs of discussion around this result. And so we followed up this study. We received funding from the COVID-19 Immunity Task Force. And what became interesting to us at the time was dose-dependent results. And so there was a lot of discussion in the literature and a lot of discussion in, well, I guess just everyone was talking about, do I really need that third dose? Do I really need that fourth dose? And then from a fundamental immunological point of view, we're interested in how much of an advantage does that third dose give you, and does it change any of the timescales associated with the immunity? The intuition would be that it does, and the intuition is that when we get a vaccine dose, I motivated our models as like our lymph system has never seen this vaccine. And so you're training the lymph system to respond to something that it's never seen before. But the way of thinking about it changes when it has trained immunity. And when you have hybrid immunity, the way the vaccine is interpreted by our lymph system, it's a lower free energy barrier to then produce more antibodies because the energy has already been put into training these cell types to respond to an unknown thing. Now you're just amplifying what is already known with additional vaccines. And so the intuition would be perhaps that more vaccines lead to longer lived responses because it's saying, I'm under more and more threat. And that's basically what we found. So we had access through the COVID-19 task force to a really beautiful high resolution data set that came from the Brockman and Broom lab in West Coast Canada, Simon Fraser University, I believe. It might have been SFU and UBC, but definitely I think Brockman's at SFU. And they had good resolution on over 100 people, at 152 people following a multi-dose regimen. And you can see by eye, this is one person on the left-hand side. Just one example of one of the individuals, and you can see their time points. So the y-axis is anti-S, so anti-spike. It's on a log scale. And then the x-axis of the left-hand plot here is time since first visit into the lab. So that's about the same time as dose one. And after receiving dose one, the dashed lines tell us the doses, but you can see after dose two, there's a huge response in anti-S or the spike antibody. And then by eye, you can kind of think that it's exponential. We love exponential models and mathematical modeling, mixtures of exponential models, et cetera. And you can kind of see it's exponential. And then after dose three, there's another huge increase. There's a decay. Well, there's a decay of dose two, and there's a massive increase following dose three, and another decay profile. And basically what we did was we wanted to remove practical identifiability issues that we noted as being present in the previous model I just discussed, and just see if an exponential fit, just a simple e to the minus beta t with a prefactor in front of that, if we fit that and we use Monolix, I should have mentioned we're using Monolix, so nonlinear mixed effect modeling for all of these fits. Sorry, that's a really important thing that I should have mentioned. Does there emergent, clear, and statistically defendable differences? And the answer was a astounding yes. And so, for example, if we do these fits on all the individuals, so all the primary series, meaning dose two and booster dose fits, were done separately in Monolix, and we collate the results together. On the right-hand side, we see probability as a function of half-life, so it's exponential, so half-life is just ln 2 over the rate, and we plot half-lives. Clinicians just are used to half-lives, and sometimes there's confusion when we talk about decay rates. And so we presented it as half-lives. And you can clearly see that there's a relatively narrow distribution around everyone who received two doses, and that the median and mean half-life is dramatically extended following the third dose. And there's also the dispersion around the median and mean also increases. So there's heterogeneity that's growing as a function of repeated doses. And so that was the main result that we published in one of our follow-up papers in 2024. And then this data had a huge distribution of ages. And so we were interested in just, okay, is there an emerging trend if we just plot our half-life as a function of age? We'd hypothesize that immunosenescence might emerge and you should see a decay. And we found a very shallow but statistically defensible decay rate of half-life of vaccine-elicited antibodies as a function of age. And because we did our primary and booster series separately, we independently found this trend in both data sets. And so we weren't using categorical covariates in Monolix. These fits aren't coupled to each other in any way, besides the fact that they came from the same people in the same data set. So we independently found this result from two different fitting a series of fits for primary series and booster. Oh, I thought I had the loss is very shallow, and I thought I put that in here, but maybe I forgot. It's something like 2% per every 10 days or something loss. It's a very shallow loss, but and so I'll just highlight or sell really quickly a paper by Professor Ian Moyles here at York, and he did a really cool asymptotic analysis of our full model that I first discussed. And he worked on picking out significant timescales associated with building vaccine immunity. And if you're into asymptotic analysis, I highly recommend reading this paper. So this is great work, but I'm not going to talk about the specifics of Professor Moyles' results in this talk. Okay, so we've motivated the use of compartmental models for understanding the inoculation of an initial unknown or unseen by the immune system antigenic response, and we've benchmarked some of these timescales, and we've tried to correlate the resulting antibody response with percent loss. And in our paper, we worked on correlating it with efficacy in a population and stuff like this. And I think we did a fairly good job of doing that, but now we want to move to what happens when there is a comorbidity with a known influence on lymphatic responses. And so, of course, we worked with a clinician at University of Toronto, Dr. Mario Ostrowski, who has long studied T cell responses in individuals with HIV. And so he also has SARS-CoV-2 vaccine studies in individuals with HIV that have controls. And so we entered into a collaboration with Dr. Ostrowski, and we're now interested in perturbations of HIV to lymph nodes. So HIV, human immunodeficiency virus, what is HIV? Well, if we relate back to some pictures that we've already seen, of course, HIV infects CD4 cells. Predominantly, it infects CD4 cells. It has other cells that it infects too, but these CD4 cells in the context of our talk and in the context of the modeling that I've already introduced, if you recall, the CD4 cell is needed to couple to a plasma B cell to share that initial antigenic information with the plasma B cell that then uses that to produce an antibody response approximately 7 days later. Is when long-lived plasma B cells do their work. And so we're interested in individuals who are HIV positive or people living with HIV. These are all Canadian individuals, and they're all on antiretroviral therapy. We're interested if we can detect differences in immune response or immune profiles or timescales in these individuals. And so really quickly, a very quick primer on antiretroviral therapy. It works extremely well if it's taken regularly, and I believe everyone in the study that we're talking about took their antiretroviral therapy on a regular basis. But it works, it's a concoction of drugs that works to arrest the dynamics and proliferation of HIV in the body. And it does that at many different steps in the life cycle of HIV. This is a really beautiful plot that I thought I would share. The budding virus is HIV, and all the different components of antiretroviral therapy are shown here, and they're all designed to target and arrest the various different stages of HIV proliferation or the HIV life cycle. And it works very well. I'll probably go over this really quickly, but these are plots of CD4 counts as a function of time. This is basically displaying viral rebound of before and after antiretroviral therapy. And so HIV, when it begins its infection process, our CD4 cells decay, and usually there's a viral or CD4 rebound associated with that before ART, but after ART therapy, antiretroviral therapy, and with concurrent usage, CD4 counts can restore near pre-HIV levels. And there's a mirroring image for viral load there. And so everyone is on antiretroviral therapy in this study. And so the timeline of this new data set is it occurred over 48 weeks, and everyone received 3 doses of COVID-19 vaccination. The separation of the population is as follows. There are 23 individuals who are HIV negative, so that's about 25% of the data set. there are a total of 91 individuals, there are 42 individuals who are people living with HIV who are immune responders, meaning their CD4 cells are near their homeostatic pre-HIV levels, basically, and antiretroviral therapy is still working. Even adherence to antiretroviral therapy can lead to immune non-responders, and there are 20 individuals classified as immune non-responders who are people living with HIV. And that essentially means that although they're on antiretroviral therapy, there's an immunity associated with the HIV could either be mutating around that or antiretroviral therapy is not working as well as it once did. And their CD4 cells are perturbed in some way such that they are now decreasing in population typically. That's usually benchmarked by a ratio of CD4 and CD8 cells to benchmark immune non-responders. And that ratio is below a certain value, the individual is classified as immune non-responder. There's one long-term non-progressor, so we don't have good stats to pick out their dynamics, and there's 5 low-level viremics, which we did our best to honestly report the stats on, but they're not really good stats on those individuals. So low-level viremic is an individual who is HIV positive and has had measurable HIV virus within the past year was the classification for a low-level viremic individual. And so the data set looks similar to the data sets that I've already shown you now, but now we have a clear classification between 2 different categories of individuals. Oh, and I forgot to mention, this is also a study of individuals that are all over the age of 50. And so immunosenescence is removed from this study in some sense because everyone is confined to an age of 50 to, I think, 75. And this came from a clinic in downtown Toronto that's a predominantly a male servicing the men who have sex with men or MSM clinic in downtown Toronto. So all the individuals in this study are male. 2 important things. So sex and age are removed as potential cofactors that might perturb differences in immune profiles. And a typical data set looks like this. I didn't do my job and label when the vaccines were administered, but you can kind of see it in my eye. There's 3 vaccines that move the anti-S signal through time, and so we can see peaks and troughs forming. This is one individual. If we look at all the individuals and just use box plots to visualize the broad heterogeneity that is present in this data set, it looks like this. And so the first 2 time points are following dose 1, and then there are 3 time points for following the primary series decay. And at each of those time points, you can see a distribution of kinetics, and you can also see a distribution of kinetics for booster decay. And so it's a very heterogeneous data set and this is antibodies as a function of time. We have anti-S and anti-RBD, and there are a few other types of data that we'll see in our modeling. And this is a cytokine response. These are IF and gamma as a function of visits, or each box is progressing in time, and the boxes are subcategorized into different responses. Did I block one out? No, I didn't block one out. So the boxes, you can see, if you just focus on blue and green, there's distributions between HIV negative and people living with HIV, and then those who are HIV positive are further categorized into cyan and purple, and these are immune responders and immune non-responders. And so for a moment, if we just focus on all the purple violin plots and progress through each of the boxes, just looking at the purple, you can see that immune non-responders have a much less IF and gamma response as time progresses. And you can also see, if you just focus on cyan, which is the immune responders, you can see that the median line of the cyan response is always greater than the median line of the green response, which is interesting. So qualitatively, people with HIV who are on ART and immune responders on median have a higher magnitude of IF and gamma response than HIV negative individuals. So sorry, blue is HIV negative. But if they're classified as a non-responder, then their IF and gamma response qualitatively drops. And so this is a clue to what we should be finding in our modeling. Went over that. This is our initial model. We've already talked about this. We need to refine it. We need to remove the practical identifiability problems that caused us issues in our first paper, and they're all reported. The random effects and everything are all reported in that paper. And we want to just start off on a bit easier ground, maybe. And we modified this model to include all the different new cell types and data types that we're now working with in the Ostrowski data set. So we still have an inoculation compartment. The inoculation compartment perturbs a vaccinated cell compartment. We're still going to benchmark vaccinated cells via that mouse study because that's going to cascade into a series of timescales downwards. And now we have cytokines that are present in the Ostrowski data set tracked through time. We have two different types of antibodies. We have spike antibodies and RBD antibodies that are tracked through time in all these individuals. And on the left-hand side, Mario was also interested in tracking RBD plus and NTD plus. And what those are proxies, are clinically believed to be proxies for a memory B cell response. So it's not memory B cells that are being measured explicitly, but RBD plus and NTD plus is thought of as a proxy for a memory B cell response. And so we include that in our compartmental model as well. Unfortunately, the RBD plus and NTD plus are very expensive biotargets to study. And so they only were able to justify the expense for people living with HIV, which is unfortunate because the dynamics are really interesting, but we can't relate them to the non-HIV cohort. And so I'll make clear of that later. And so our first result, if we remember from the cytokines, there were clear differences qualitatively, and there are clear differences in the fits. I picked a really bad color grade scale, and so I apologize for that. I apologize for that every time I give this talk. It's published now, and we published this in iScience in 2023. And so this top plot is the population fits to people living with HIV, people without HIV, and then the immune responders and immune non-responders. And we fit them all simultaneously in monoliths with categorical covariates to try to pick out statistical differences between these categories and fits in all these parameters. And the gray regions are just so you understand, there's a huge heterogeneity in when the doses were administered. So the gray regions are the dosing intervals for the primary series, the first region, and the second gray region is the dosing interval for the booster series. They kept the visits to pull the data consistent, but the time of dosage is inconsistent. So that is an issue when we're trying to think of dose-dependent dynamics for some of these time points. But we're able to find a result that really mirrors qualitatively what we can see in the data that emerged from our fits, our production rates and decay rates, or production rates that match the qualitative trends that we noticed. And we were able to benchmark the dispersion of these kinetics as a function of the classes that we're interested in. And so it's interesting here on the left-hand side is HIV negative, which has a fairly shallow dispersion in interleukin production, and it's an exactly mirrored result for IF and gamma. So I'm just going to show one of them. But when we model fit to the HIV positive, there's a huge range of production rates, and we can see that range is being pulled up by the immune responders and being pulled down by the immune non-responders. And so there's a kinetics that we can associate with these T cell frequencies that are being elicited by these cytokines, interleukin 2 and IF and gamma. And so this is one of the results that we contributed to this paper. And so basically what's happening here is that we think that immune responders have elevated follicular cell and helper cell responses in the lymph nodes at baseline, and then they're dramatically reduced at baseline for immune non-responders. And then Mario believes that this is called T cell imprinting, and there's a whole body of literature around T cell imprinting. It has a bunch of different names if you're in the HIV literature, but the one that Mario goes with is T cell imprinting. And this is evidence for that, he believes. And what's interesting is that although there's obviously huge differences between T cell responses, we were not able to find basically any difference between antibody responses. So IgG spike, IgG RBD, even if we perturb the structure of the model a bit, all the data between people living with HIV, people without, immune responders, immune non-responders. The spike decay rates were very consistent and very similar. And we looked at a function of dosage here again. And so, for example, they all had roughly half-lives of 29 days for primary series and 80 plus or minus 22 days for the booster dose. So a huge range on that. But the dispersion and the median values were not affected by the category of the individual. And so that's fascinating because the cell types that are associated with mounting the response are definitely different, but there is some sort of rescuing happening physiologically such that the antibody responses are consistent. And so that was a big result for this paper. Also, this result has been refound in a different paper by Brockman and Broom, actually, and another statistical paper has shown that antibody responses in people living with HIV who are on antiretroviral therapy are consistent or similar to non-HIV controls. And so there's something really interesting physiologically happening there. And it's now been discovered in the context of COVID-19 vaccination. This has been also published by other groups. And so finally, the final story I'm going to talk about, and this is unpublished work, although it's now ready, it's basically ready to go now. Mario extended this data set from 3 doses all the way through to 5. And so everything that we've talked about so far in this HIV study has gone to 48 weeks and 3 doses. And then Mario also extended now this data set by following a subset of individuals to dose 4 at 78 weeks and a dose 5 data point at 104 weeks. And at this point in, I guess, my career, my tenure in Jane's lab, I became really interested in machine learning. And I was doing some machine learning stuff. And I thought, well, we have so much data and we have already sort of classifications built into the study. So there's a non-HIV control and people living with HIV. Well, machine learning is built to learn about what in the data set contributes to these classes, and it wants to learn to pull these classes apart and figure out if it can do that and learn the signatures. And if we apply this to an immunological study where everything is continuous data and this is all blood-drawn data, we can learn about what's going on in an immunological signature amongst these individuals and see if there's certain features, immunological features that emerge as really important at certain points in time. And so that became the motivation for this next study, which is using machine learning with all this data that I've been talking with you about to understand what's different about these people with HIV and people without HIV. And so when you get into machine learning, it's best practice not to just jump into some complex AI model that has deep neural nets because maybe it always compiles, which is an issue with machine learning if you've ever forayed into it, but you don't know if you're overfitting patterns and you don't know if you're getting meaningful results. And even if you look at your ROC curves, you don't know if you can necessarily trust them. And so we took a really structured approach by starting with simple methods, so simple clustering methods, like principal component clustering, all these individuals with all their immunological data, do they form distinct clusters and principal components? And we look at the mapping of principal component 1 and principal component 2. And the answer was interesting. It wasn't definitive. So we'd really like to see HIV negative cluster meaningfully out of an HIV positive cluster, but they subclustered within an HIV positive cluster. And we can see the HIV negative individuals form a tighter cluster within a very heterogeneous HIV positive cluster, and the ellipses are 90% confidence ellipses, and identified, that becomes interesting in this story, some far outliers in this data set, ID 70, 72, and 49. And we're interested in this vast, I would say vast because there's 63 immune features. And for an immunological study, that's a lot of data. It's a very expensive data set. For a machine learning study, it's pathetically small. I get that. But from an immunological point of view, it's a really huge data set. And so we didn't really know what to do with this result. There's clearly subclustering. And then we went to linear discriminant analysis and found, okay, with the linear model that's built to drive responses away from one another, there's absolutely clear separation. And that motivated us to use a random forest approach, basically, to to apply to this data. And random forest is really nice because you can write code to lift the hood on that machine learning algorithm to figure out what it's thinking and how it's making its decisions. And so our paper became a fairly long paper using supervised machine learning approaches to understand classes in this classes between people living with HIV and people without HIV with all this immunological data. Really quickly, if you haven't read about random forests, it's a really powerful, very fascinating algorithm to assess classification criteria, but it's also really nice because you can lift the hood on a random forest algorithm and look at all the different trees and look at the ways in which it's weighting all the different features. And you can see why it's making the decisions that it's making. And we're interested in looking at why did you make certain decisions for certain individuals that are always outliers or always misclassified. And so, for example, if you have someone that's HIV positive, but their algorithm always thinks that they're HIV negative, and we have two of those individuals, the answer is, well, why do you think that? Like, what about their immune profile is leading you to think that they're always HIV negative? Conversely, someone who is HIV negative, typically it learns that class very well. Like our AUC is like nearly 1. It's an incredibly high population level classifier, incredibly high correct classifier at the population level. But then we can say, well, why does this one individual or two individuals who are HIV negative, you always think they're HIV positive? They have an immune profile that looks like an individual who is HIV positive and on antiretroviral therapy. So our entire paper becomes a story about classification and learning about outliers and using machine learning to benchmark interesting immune profiles that don't fit typical signatures. Yeah, our ROC curves are really nice, but they depend on the type of data set that you're using. And so, for example, cytokines turn out to be a fantastic classifier, as we've basically alluded to in the previous few slides, that they're meaningfully different between the classes, and serological IgG is not. And we just reconfirm that result to benchmark ourselves in what we believe we already know, and that is emerging as true in random forest. So if you only use antibody data, you get with the red curve here, which is sort of a weak classifier. It has a mean AUC of 0.74 and a huge distribution of ability to classify. Whereas if you only use cytokine data, you get a very strong classifier. In the main context of the whole paper, we're using all the different data types, but we do consider subcategories of the data. I was pretty sure I'd be running out of time at this point, so I didn't add any more plots to show what it looks like when we lift the hood, because they're really complex multi-dimensional plots. And I'm at 50 minutes. And so I'll just end with a quick advertisement for a final paper that we're working on with Jessica Conway at Penn State University, where she has collaborators that have collected data for SIV female macaques that were inoculated with SIV in their female genital tract, and then they tracked that SIV through time, through all the different tissues. And so you have a data set where you can learn about network of SIV spread within the first 14 days of infection. And we're using Bayes' theorem to build these networks and understand SIV spread. So if you're interested in that, drop me a note. We have a lot of cool things going on there, but I think I'm running out of time. And so I will end with, yeah, I was pretty sure I ended there. I'll end with our acknowledgements, which I've already gave, and take any questions.
Okay, thank you, Chapin. And as I'm online, I will leave it to the people in the room to moderate the question period.
Can I ask a question from the floor?
Go ahead. Yep.
I think it was a fascinating talk. I'm from Edmonton. It's great to hear this wonderful work going on. The work at the very end, you talked about the machine learning results, and you sorted some results. Was that on a held out data set, or is that part of the testing set that you were showing?
On the ROC curves?
All the results you showed.
All the results I showed was on the testing data set. Sorry, not on the training data set.
You did split like 5-fold cross validation or something.
Yes. Oh, we did. Yeah, we did KV 5-fold cross validation, and we shuffled the data thousands of times, and then we had external we had a holdout data set and everything, and we did
Okay.
all the proper things we're supposed to be doing, I think.
Excellent. Thank you very much. That's very impressive.
Yeah. Yeah, that was on the testing data set after all the different folds, we did 1,000 different iterations of 5-fold KV on, and that's the results that I was showing for that the test set.
Thank you. Any other questions from online or in the room?
I was wondering how energy of the cell is involved in this model, because in the cellular and molecular level, what is the initial states of the energy? I mean, the time scale you have applied for your data set based on the ODE model. And
So I heard, I caught some of that. Was the question about initial conditions, or was it about, I forgot, I just caught I caught parts of that.
yes, it was about initial condition. I'm talking about the energy level of the cell, cellular, and the time scale mostly. The time scale for the cellular level, the energy is somehow different for the vaccination period as you had it for 30 days, 45 days.
Oh, I see. I think I'm not sure. Energy level?
Yes, energy level.
Oh, I'm not sure. I'm not sure.
Yes, I'm talking about the energy level and time scale.
Okay, yeah. In our compartmental modeling.
Yes, for the cellular and molecular, how the time scale will be involved. Because you're talking about two different time scales. Your model OD is the time macro time scales, but for your molecular and cellular, the time scales and energy
Oh,
levels of cells will be different.
okay, yeah, okay, I understand it now. Yeah, so we have compartments that are associated with the net response of a cell type like plasma B cells and CD4 cells and CD8 cells. And I also talked about we have IF and gamma and interleukin 2, which are each molecules or proteins. And there are clearly different timescales of interaction between molecules and proteins and cells. Is that what you're referring to?
Yes.
Yeah, okay, so it's a bit misleading because IF and gamma and interleukin 2 are ELISpot results. So what they're doing is they take blood from an individual and they're using spike peptides to elicit a response, and they're looking at the number of T cells that are producing IF and gamma, the frequency of T cells that are producing IF and gamma, and they're looking at the frequency of T cells that are producing interleukin. So it is actually a cellular compartment or a cellular measure that we're using to infer a timescale for a bulk cytokine response. Does that make sense? Yeah. So I just didn't do a good job of introducing that.
Okay, thank you. Well, seeing that it's 11:30, I'll move it to the room for the closing remarks.
All right, thank you very much today for coming, and it was excellent presentation, great work. Thank you very much for sharing your work, and yeah, we'll see you in a month again. Professor, do you have anything to say? Okay, in that case, thank you everyone. See you next time.
Thank you very much.